There we go.
Nice.
Okay.
Okay, full wide, Wayne.
Full wide.
I turned it to 0.
It's not in the jar yet.
Got to keep it on at
Yeah.
least 30. Nothing in the jar yet.
Mm-mm.
You can go 70% maybe.
Oh, a little came. Got a little bit of more rubble. Come on.
Maybe give it 100. Full beans.
Full
See if we can pull through there.
Is it worth it to try and send something else through, or does that
There's more
clog it
purple
up?
over here.
Just on the bottom too.
Can you zoom in a little bit, please, Wayne?
Zooming in.
I think there's some purple right there.
Yeah.
Mm-hmm.
Come out, just a bump. Yeah, good.
Still on 100.
Sure.
Full juice. There's another little piece like right in front of the porch.
Can you stop the suction for a sec?
Yeah.
Okay, now do 30. I might have had some cavitation going on.
Yeah, it's not.
Flow's pretty weak.
I'm seeing no suction.
Yeah.
We got a little bit of movement in the jar.
No, go to 0 again. Okay, 30 again, maybe 40. I think we're done slurping for the day.
Nothing.
We need a reverse.
Yeah, need a different style of pump and everything then. Okay, that is what we get today. Okay, you can 0 that.
Full wide.
0% on the suction.
Look at that brittle star on the move.
Mm-hmm.
Wow.
So somewhere in the tube there is that sample. It makes a couple turns on the way from the bottom bar right there where we're resting on the seafloor. It comes in, turns a 90 and goes up vertically. You can kind of think about it like moving up to the shoulder of the vehicle, rising about almost 6 feet, and then it takes another turn and goes kind of across the light bar, so almost like across the forehead of the vehicle and into the jars. So somewhere along that journey, got that brittle coral hash clogged up that tube.
I'll put it back to flush jar just for good measure.
Interesting. Good question. So with it being 5 degrees Celsius down here, why don't the fish get hypothermic? Fish are not endotherms in the same way, or let me say this, most fish are not endothermic in the same way that mammals maintain a higher internal body temperature than their surrounding environment. That's really important for lots of reasons, like our metabolism, our ability to digest, and just our thermoregulation. We are, humans are really only tolerant within a fairly narrow range of temperatures, the way we've evolved, but a lot of these fish are just maintain the same temperature. What's unique about some fish is that they do have the ability to elevate their body temperature in certain ways. So some sharks and one of my personal favorites, tuna, actually have the ability to, they have a countercurrent heat exchange that happens near their near their gill arches. So taking some of their expended muscle energy and exchanging with a hot to cold temperature flow, and they actually can heat their brains and heat their eyes, which allows sort of a quick, more rapid reaction and an increased amount of blood flow, which is an adaptation that makes these predators like a little sharper and a little faster than the things sort of in their environment. So tuna can heat their brains and their eyes even if they're up in Arctic or at Arctic waters. Not something they need as much if you're down in tropical waters, which they also range to. But that ability to elevate body temperature is not the norm within fish. So these fish are just kind of all the way through. I would expect any of these Oreo fish, any of these conger eels, anything that we've been seeing down here, if you could take their body temperature, it would be just the same as the surrounding environment. Trevor, the viewers want to know, what do we do when the slurp gets clogged like this? When we get back to the surface, how do you fix it?
We tend to fill it with water, then dump it on the deck, and it kind of, the suction in reverse pulls it out. So we don't have a way. The pump on board Herc is only designed to run in the one direction. It can't run the other way. Mm-hmm. So in order to get that flow in the other direction, we just pull the slurp hose out, fill it with water, and drop it on the deck, and that siphoning kind of thing sucks water all the way back through and pulls all the bits out. And failing that, we can take it apart. The hose is in multiple sections, and get pokey, poke stuff through, poke it out. That's pretty much it.
That'll do it. That's it, because it works. Awesome. Thank you.
That was one of my summer jobs, was on a split hull hopper dredge. And it had these giant arms that would dredge Mobile Bay, and it would just scrape everything up off the bottom. And these arms were large enough, and these pumps were large enough to process a regular car tire, which a lot of the boats used as bumpers, but not the 18-wheeler tires. The 18-wheeler tires would get wedged into the blades of the impeller in the pump.
That's a massive pump. Holy cow.
Since I was the intern or the summer job, I got the dirty job, and they would send me into the pump. I'd have to crawl up through the arm and get these tires out of the impeller.
Oh my gosh.
And trust that they were not going to turn it on while I was in there.
What were you sucking up? Where were
Tires.
We were dredging Mobile Bay to make sure the bay stayed navigable.
Shipping channels open?
Yeah,
wow.
yeah.
And there's a lot of 18-wheeler tires in Mobile Bay.
There's a lot of everything.
There's a lot of tires in any navigable waterway, for some reason, where we were dredging. Anyway.
Wash out of harbors, wash down.
Surprised that wasn't part of one of those dirty job episodes.
Yeah.
That would be a good one.
I tell you what, I went back to school and I had my best grades ever.
We use big dredge pumps to remove invasive algae off of coral reefs in Hawaii. With a big barge with underwater vacuum cleaners, kind of like the slurp sampler on steroids. Our hose was about 5-inch diameter. So the in-between size between Wayne's story and our story here. We'd vacuum that up and pull it off because it was growing so much more quickly and could shade out the corals. The Hawaii Super Sucker barge.
Bunch of bottle
Are
brush
those
here.
all crinoids?
Yeah.
Right there was one sea star down below, but
It's your field of crinoid stream, Miles. Woohoo! Do you just want to frolic through there?
Romp.
Romp.
I made a Venturi dredge working on a lake.
Yeah.
Yeah, it always got clogged up with sticks, but
It was
did its
Sticks.
job.
Nice.
With what?
A Venturi dredge?
No, it got plugged with what?
Sticks.
Sticks, not steaks.
Sticks. Yes,
Just
no meat involved.
Then
what
I'm
about
out.
flesh?
Crunch, squish.
I'm one of the only people I think to date. I don't know. I hope nobody else has, but to be vacuumed into the super sucker was my mistake.
You were what?
My arm got stuck in it.
Stuck in what?
In the vacuum dredge, the Hawaii super sucker.
You're supposed to be behind the nozzle?
Yep. Well, when you're clearing it out, and the trouble is when you're wearing a wetsuit, because then it can form like a nice seal. Because my arm is not 5 inches diameter, but with enough wetsuit, it was. And when I left that job, they gave me an award. They made a plaque where they took a Barbie and they jammed it down in a garden hose, like all the way down in there, and then put it on a plaque and awarded it to me on my last day. It's really sweet. Love those. I love those guys.
I'm not so
Just
sure
tears.
that's laughing with you.
Oh, it was. I mean, yeah, I trained a lot of that team and been with them a long time, but still got that record.
Did that increase your wingspan?
You know, that is a really good question. Yeah, I thought it had broken my arm. I ended up in like a sling, but mostly with like a giant arm hickey for a lot of it. It was pretty rough.
I hate those.
Yeah, it's real weird. It's real weird. But yeah, dredges of all sizes. Did you guys know that most of De Beers diamonds are actually dredged from underwater? Do you guys
I
know
did
that?
not know
A
that,
lot of
no.
diamond mining is now happening underwater because they run down in rivers. And
Yeah.
so, yeah, massive diamond mining
Especially
collected
in
from
South
underwater.
Africa and
Yeah,
Namibia.
Namibia,
Mhm.
yeah.
Is it roughy?
It is. So that's super cool. I didn't know anything about that, but I highly recommend. There's some really good coverage. Seems to have kind of alerted onto the global stage a few years ago, although it's been happening for a long time. There's a story all about diamonds in the deep, gems being mined from the bottom of the ocean from last year.
How deep are they?
Oh, they're not very deep at all. They're working near shore.
It's got to suspend a lot
You
of
know,
sediment.
oh yeah. Yeah, they can't see anything. They just drive along the bottom from the story. So, you know, shallower than 200 meters.
Yeah, Steve says the oxygen's dropping rapidly here.
Why would that be, Steve?
Stop breathing so heavy, guys.
Jake is doing his part by holding his breath.
Can we zoom in when we get a chance?
On what?
In anything, just to see if...
See if the zoom still works? Roger.
So this is
You
the
all right, Jake?
Yeah, you can zoom whenever.
All right.
Just want to see what's there.
Oh, is that purple?
I want to see if it's all dead.
Brittle stars?
Brittle stars
crinoids,
and crinoids.
Purple.
There's some of that purple. Yeah.
It's amazing when you zoom down, you just don't think there's a lot there, but you zoom in, that's a lot.
Man,
For a
it
very
comes
small
alive.
area.
I can't hear you, Tim.
Oh, just saying how amazing it is, how much life there is when you zoom in.
So I can see some of these white shells.
There's
What
a
are those
shell.
sponges?
That's
Or
like the first shell I've seen.
yeah.
Hard to see. Yeah.
There, that's ideal focus.
So the oxygen minimum zones are caused in the open ocean. Steve, back me up with some more knowledge I drift into the rhubarb here, but oxygen minimum zones form, well, let's start at the surface. When there's upwelling and there's a fresh water, nutrient-rich water comes to the surface and gets to the photic zone where you can really have a big bloom, a kick-off of algae up there. You can generate a lot of primary production, and those things start to sink. So whether they are the actual phytoplankton themselves have a lot of adaptations to stay up in the surface waters, but if they're consumed and processed, another stingray.
Yep.
So as that
Zoom in.
Okay, zooming in.
Yep. Yeah, can we get a picture without the lasers too? Well, I guess it's...
He's far away.
That's great, right there.
I'll tip down your Argus cam, help you out there.
Oh, great shot on Argus.
Let's see if we can perfectly frame. Perfectly
nice
frame.
lady stingray. No claspers.
That's nice distance shooting.
That's so cool. Plesiobatis stingray.
Can you write that in the chat log?
Of course I can.
Okay.
Stand by. He's coming center. There we go.
She. So stingrays, like sharks, have claspers. You can't see them there. They'd be right along the tail.
Okay,
they were there.
wide.
It's
Wow.
obvious sexual
Thank you.
dimorphism, so you can externally tell the difference between males and females.
Nice job, Jake.
Yeah.
Nice
Yeah,
one,
great job.
Jake.
Chasing him down. Chasing her down.
Chasing her down.
Once you get back out front, I'll camera up again.
So back to oxygen minimum zones, after a bloom in water, and whether things are consumed and, the marine snow, the poop of zooplankton starts sinking, or whether the phytoplankton itself starts sinking, there are bacteria also in the water column that consume those things. And bacteria consume oxygen while they're consuming food. They respirate, or they respire, respiration. The typical parallel process or reverse process, photosynthesis and respiration, like we think about shallow water plants, but here it's separated, photosynthesis in the primary producers, respiration by bacteria and consumers. And as they are consuming everything that's come from this bloom, they're using up the oxygen. So it's pretty standard to have a band of lower oxygen water, what we call an oxygen minimum zone, beneath the productive waters. And depending on where you are in the world and what oceanographic conditions are occurring, whether there's upwelling or mixing, the oxygen minimum zone can move up and down based on those big kind of global scale oceanographic parameters. So we can see that happening. Steve's watching the oxygen and seeing it drop as we're rising up into this band. That's important to know because it impacts the creatures that...
Stay. Yeah, thanks.
Impacts the type of animals we can see. There are species that are more or less tolerant to these naturally occurring oxygen minimum zones. And a few people writing in knowing about dead zones. Those are not the naturally occurring type of, it's the same mechanism though. Big bloom, lots of consumption, eating up all of those bloom nutrients. Where the system starts to, where you'll see these in the news and it starts to be more popularized and more damaging is when you have like a lot of fertilizer, for example, dumping into the water, creating these blooms that are sort of out of proportion with, huge boom and bust cycles, I guess you could think of. And in those huge bust cycles where the oxygen's used up by bacteria, you get widespread dead zones. And those are what make the news off of in the Gulf of Mexico, off of the Mississippi River, in the Mekong Delta, lots of places where there are major river outflows around the world. Tim, are you on the question feed? Do you have it open back there?
I can't open it.
Okay. There, you have a special note from Logan in there.
Oh, hi, Logan. Okay, so from Logan. So rays, on the reproductive organs on rays, we call them claspers. And yes, they have 2 claspers. They're one on each side of their tail, but they're kind of tucked in right underneath the tail. So that is true. Your father is correct.
I think the one we just saw I didn't see any claspers. Did you see any?
I did not see any claspers, no.
Lady Ray.
Mama Ray.
Ah. Super cool having people follow along from all over the world, but also extra cool when, you know, friends and family tune in. So thanks everyone for being here with us. I think it's one of the things I like most about my job. I think if I want to trace it back really to how I got interested in doing science communication, it was the idea that I just wanted to share the adventures I was having. I wanted to share, you know, the excitement that was happening in my life and in my career with, you know, with my friends and family who, by and large, are not anywhere near an ocean. And so it's really, really cool when we get to just hear from folks about where they are, where they're participating with us, you know, what this experience, you know, what questions it brings to mind for them, what curiosities it brings alive, what career pathways or passions for learning that that it helps light off, because it certainly has done that for all of us. You know, we're out here because we love to keep learning and we love what we do. And it's just cool beyond cool that we are able to use telepresence, this system of technology, to share it with researchers all over the world, but also just folks that are interested and passionate about the seafloor with our next generation of explorers.
Want to take your bubble?
Yeah.
Little spiny sea urchin off to the left there.
Yeah. So interesting that makes sense as we're going into this oxygen minimum zone, but just to sort of have a stark transition in the amount of coral here.
If you want to zoom.
Yeah, let's go. Can we zoom in on that urchin?
Yeah. Which,
Yeah.
Upper right.
yeah.
It recently had
now.
its hair bleached. There it
There
is.
you go.
Ooh, nice urchin.
Yeah, I saw another one like that just below.
Yeah, I want to see that.
It's a scary one.
It's really cool.
There's something growing on it too, it looks like.
Get the not quite focused. Yeah, there's some hydroids on one of the spines.
A lot of that stoloniferous coral here too.
Mm-hmm.
Yeah, Steve calls it a pencil urchin.
Very cool. There.
Might be good to give that camera a little bump up and get the slurp hose out of there.
Oh, wrong way.
It's true.
It's the Jared urchin. It's nice.
What is this?
Another big fish.
Cuskeel.
Yep.
Our rat tail.
Probably have to be fast because you're starting to fall behind.
Ooh,
Okay.
It's a slightly different one.
photophores on the head.
Yeah.
Yeah.
This is the one with the long snout.
It looks mean. It looks fast.
Fighter jet. Look at how fast he's going. Good observation. That's awesome.
Look at the Oreo fish.
I love it. Look at his
Oh,
eye. Oreo fish. Yay. Yeah, it just looks like it tumbled down, right? You can really see that tumbling there. Fell right off that side. Thanks, Steve. A secondary influence on how low the oxygen here is the upwelling of poorly oxygenated deep Pacific water to the surface. So some of that water that is coming up is not necessarily, you know, it has been deep water for a long time in the Pacific on this conveyor belt of different water bodies that move around the world. So not coming up fresh and full of oxygen. Good point. Thanks for the backup.
So we got another little micro plateau up here.
So these bony fish we're seeing don't have claspers. And it's kind of interesting that it's two different forms of reproduction. With your sharks, your rays, and your chimeras, those are your cartilaginous fish. They all have claspers, which allows them to have internal fertilization. So that also means they give live birth. So there's a lot more nutrients coming from the adults and allows the juveniles to be larger when they're born and have a greater chance of survival, versus a lot of our bony fishes, I guess all our bony fishes have, they produce gametes, so sperm and eggs, and they're spawning, releasing those, you know, hundreds, millions of gametes into the water column, which then have greater dispersal, so they can drift off with the currents. But there's a much higher rate of predation.
A little hot there, Wayne.
So you might have 100 offspring,
Nice.
but only expect one of them to survive.
That was a great explanation of that. Thank you. We don't expect that fish at this depth are blind. That's not our understanding. They do have eyes and different types of photosensitivity to light. They are adapted for an environment without light. We are bringing all the light you're seeing with us. This would normally be a totally dark environment. As far as long-term studies, there are not a lot of them understanding the impacts of light in the deep sea, but it's one of those things that we would be unable to function down here. You could not fly ROVs safely without bringing your own light. There are lower light IR, some infrared, some just low light cameras available. But as far as producing video like this, you would not be able to see any of the landscape. If you want to see bioluminescence, it almost looks like a dark field with just bright splotches in it. So there's trade-offs, to produce video data as a or high definition video streams as a data product. You need to bring enough light down for cameras to function. So, we do understand we're getting a survey of the environment, moving quickly through this area and understanding what's here in an area of management as a benefit for the fact that we are, exposing landscapes to light that they don't usually see. Also understand that we're making a baseline that while we hope that more dives would come back here and a lot more exploration, with so little of our world's ocean explored, and currently, the scale of resources that are devoted to exploration, it could be years, decades, even longer before anyone returns to these sites and does any type of hypothesis-based research or further exploration. So try to gather all the data that we can while we're here. Many of the animals that are described from this area were described, or even in the 1800s, via trawls from some of the first exploring expeditions or Western exploring expeditions that came through the areas. So, understanding it could be hundreds of years until someone returns to this. Obviously, we hope not. We hope to be in an era of exploration where we start spending a lot of resources and devoting curiosity to understanding our oceans and our planet, but it's entirely possible no one might be back for a long time. Somebody's got it like pretty close to their nose. I'm not sure who it is.
Lots of crinoids here.
Yeah, lots of crinoids. These yellow crinoids are so cool. Oh, Plegathuria.
Some bottle brush here again.
You're muted
Or,
too, Wayne.
or are they different? No, those look a little different. No, they're the same bottle brush. I didn't see the crooked stem.
Did you see bottle brush?
Yeah.
Did you see the Plegathuria, Suna?
No, but I heard it.
Cool. I really like this fish. This one makes me happy. And the banding is so unique. It makes me wonder if it's always there or if it, they have some control over the banding.
Can we find this fish?
Yeah, let me see what I can do.
I found it. There it is.
Oh,
Good
thanks,
job.
Trev.
Yeah. Glad to help.
That was quick.
It should be some kind of rat tail.
Mm-hmm. Yeah. Open up.
That fish looks so hurried.
Important Oreo things to do.
Don't mind me.
All right, Jake, it's about to get steeper.
Okay.
You up for it?
Looks like we're some of these precious coral.
Yeah, he's up
Yeah.
for it.
Let's hope so.
Colorinchus.
Colorinchus tokenensis.
I think so. That's what we're here for. Passing the torch. What's that?
I think he's getting the hang of it.
I think so.
That's awesome.
I think
You're
he's
doing
born
great,
with the hang
Jake.
of it.
We call him the natural.
The prodigy.
Still not breathing though.
Really hard to talk and not breathe. You're doing great, Jake.
Anatomy.
Yeah. Crab.
Crab, yay.
Oh, yeah.
So we don't have any mid-water or water column exploration planned, a deliberate water column survey. We have been noting pelagic species as we see them and as we ascend and descend through the water column, but no dedicated mid-water transects on this expedition. There's a lot of interest in that. I mean, it's yet another piece of the oceans that we don't have a really good handle on the species distribution or some of the even oceanographic and chemical just parameters. And so lots of exploration effort happening there and also lots more work to do. Viewers want to know if we've seen any slime stars. We haven't seen one this watch yet. Doesn't mean we can't, but no chicken skin slime stars yet.
What was that, a slime head that we
Yeah,
saw?
slime head, yep, roughies.
Did you see the Argus occlusion?
Oh, yeah. Look at that.
Oh, yeah.
It's getting steep.
Here we go.
Only 600 meters, though, so ship control is aces.
600 meters?
Up depth.
Oh, yeah, yeah, yeah. There's not much layback, and we can stop fairly quickly.
Mm-hmm. We're still going 0.2?
Yep.
Cool.
What's the deal with these rocks? Want to tell us about these rocks? If this was a basalt, that would be a crazy pillow, but it is not a basalt. Want to talk to us about it, Rob?
Yeah, these are part of the the former carbonate reef that was probably farther up on the surface. It looks like they may have fallen down, slumped off the edge. And so they're all just, you know, broken up bits of coral and calcium carbonate that were formed as the atoll became more of a guyot. Actually, it's not a guyot here, but it's just a fringing reef with carbonate. I think a lot of the stuff has fallen down and slumped down off the side.
What's our oxygen level at? We're now
46,
so
Just a minute.
45 and a half.
Yeah.
what was the low when we went through the zone?
I think it's still dropping.
It
Oh,
used to be
is it?
around 68. Now it's 45.
Okay.
Still dropping.
Is that micromoles per milliliter?
Per liter.
Per liter.
And we're at 629 meters.
And what would surface water be? What would freshly mixed oxygenated water be? Maybe a Steve question. Help us, Science Steve.
Around 200, he says.
Awesome.
Whoa.
And we're at 40 something.
We started seeing corals again.
Yeah, here on the vertical. Anemones don't seem to be, or these seem to be tolerant, more tolerant of the low oxygen.
Can we zoom in one of those corals? Is that possible?
I don't think so right now.
Okay.
I gotta get up to the top of this thing.
Oh, little anemone village at the top. You can see the marine snow coming down to them. It's a great place to be an anemone.
I do like the choice of following this crevice all the way up.
Ridge, hogsback.
Really interesting.
Look at how many
that
of them
one?
are clustered right there.
That's a good
What's that?
cluster.
Said it's a hogsback. Not a
Oh,
valley.
sorry, that's not what I heard. Yeah.
That's awesome. So anemones can reproduce by budding, which seeing that many of them that close together makes me curious if they bud off from one another. Look at them. Oh, they just look like pom-poms or fireworks. These are anemones are a favorite of mine.
I just love the zonation changes we see as we come up shallower.
Mm-hmm.
This is our last big climb. Or the start
Ooh,
of our
crab.
last big climb.
It's a cool crab.
Where?
Left, bottom left.
Oh, yeah.
Looks like we're probably in a better spot now for future zooms.
Okay.
Okay.
There's a crab. Just on cue.
Ooh, Oreo fish.
Gyrating holothurian.
Yeah, so
Well, there's something
A yoga cucumber.
floating
yeah, you.
Yeah.
close.
A little different look.
Yeah,
Yeah.
did.
More hooded.
I don't think we need the crab.
Pentala urchins.
Mm-hmm.
I'm not a scientist, but
Yeah, Steve was asking for the fans, if zooms
Yeah.
on the fans, if we can see them. Find another. Is that Coelurhynchus? Yes.
The banding
I'll
is
give him
good.
a zoom. You ready?
Ready.
Coelurhynchus.
Nice framing.
Nice focus.
Yeah, there's some small corals right up there
Coming
too.
wide.
Yeah, right above the lasers, there's some coral there. Get a zoom on that.
Did you hear that, Jared? They asked for a zoom on
Ooh,
those
there-
corals?
There's
Where are the
crab.
corals?
To the right of the lasers, down now.
To the right of the urchin, they're white
Yeah, down.
little fluffies.
Oh, yeah.
White fluffies,
Mm-hmm.
right of the urchin.
Oh, I see him straight down.
They're not the ones that Steve wanted
good for a
originally,
zoom.
but just gonna
You
look
ready
at
to
these.
push in?
Yep, ready.
Pushing in. Are those the white fluffies?
Yep, that's the ones I'm looking at.
Sponge? I don't see polyps.
I think those are sponges.
Yeah.
Okay. Okay.
Sorry.
White fluffy
Yeah.
sponges.
Coming wide.
Glass sponges.
Okay. No interest?
Furry there.
Okay. Moving on.
Steve, do we know how long anemones can live? I don't know the answer to that. Nope. Awesome.
Steve says nope.
Steve just says nope.
Does anyone know?
I don't know how you would age an anemone. I wonder if they I don't know. It's not like you'd be integrating any hard
Right, there's no ear bone. What's that one called?
otolith.
Otolith.
Even the way you identify anemones can be really difficult. You identify them by partitions that are internal to their body. So in order to identify anemones down to species, you need to collect them and cut them open. And know what you're looking for. Taxonomy of anemones is certainly a specialized field. The coral question of how the corals are seeing reproduce. Corals are broadcast spawners. They release gametes into the water. So the corals that we're seeing down below, and a few of them that we're seeing here, would release sperm and egg into the water, and then gametes would combine and eventually settle down, hopefully land on hard substrate, because they need hard substrate to anchor to. So a bit of a lottery on whether or not where you're going to land and whether that is an environment that is I guess just possible for you to grow in, has the right current conditions to bring food to the polyps to filter feed, and the right oxygen availability to grow in the tissues. So definitely more of a lottery approach. The coral rubble, it's not possible to know how long it has been dead, although the stuff that is bright white, more recent than the stuff that is covered over more with additional marine snow or bacteria growth or hydrozoan growth. Our co-lead scientist Steve Oskovich estimated that we're looking at the decades to hundreds of years scale on most of this rubble. And we've been seeing rubble all throughout the climb, so interesting to know that there was enough growth, enough coral abundance at different points in the seamount's history to have created a really stunning amount of coral rubble for hundreds of meters below as it tumbles down or is pushed down by currents. Or as colonies used to be at different depth ranges along the seamount.
Permission to zoom?
Yeah, these holothurines look a little different down here too.
Cool.
Well, there's a little holothurine down below too. Stubby one.
Yeah, he's got one stubby.
Yep.
You should see the other guy.
Yep.
Stubbithorian.
Thank you. Full wide.
Hi, Dr. Chris Kelly. Thanks for writing in. There are also deep sea corals that are brooders, which is great to know. Another strategy of reproduction. Brooders fertilize the eggs and develop planula larvae before they are released out of the colony. So that the two approaches would be release sperm and egg into the water, have them combine in the water column and land, or fertilize the eggs internally, develop into a planula, and then release those out. Thank you so much for the help, Chris. We really appreciate it. Starting to see a few cool glass sponges here. We're certainly into the oxygen minimum zone if you've just tuned in.
Can we zoom in on those little fish down on the bottom right?
The-
Keep seeing them. They seem like they're maybe McTophans.
Bottom right. Where are you looking?
Bottom up, just went out of screen.
One in still, right out
You got
now.
time?
Or just next time we see one of those skinnier fish, just
A stingier fish.
kind of a
Skinnier.
small and skinny.
Skinny.
Yeah. They do look kind of like McTophids. I was thinking of the same thing. I would be intrigued, but also excited to start seeing them.
Next cliff is in sight.
Now it's 600
Cliff in sight.
meters.
60 meters. 55.
In shouting sight.
50.
Question coming in from the team. What career milestones do we hope to achieve? I think Jake is doing it right now.
Just gonna check all the boxes, then leave.
I peeked.
Yeah, I know.
I've peaked.
Oh,
Summited.
Yeah, pretty awesome. I mean, you were flying before on a slow sloping sediment kind of sand ridge, and now you're flying up a cliff.
And chasing rays and
big
crabs.
crabs.
Yeah.
Crab wants to fight you.
Yeah, that crab wants to fight.
Come at me, bro.
This is my mountain.
There can only be one.
Come at me, bro.
That's about the end of your leash there.
So, Jake just got on board. How long ago, Jake?
12 days ago?
Nice. And had you ever flown an ROV before?
Never. Never ever.
And what you doing now?
Yeah,
I am flying an ROV.
Yeah.
What is that?
Well, we're
Is
not
that-
we're
is
not
that the one you
broadcasting
want?
the control van camera, so we can just.
Nope, that's different.
that's different.
Yeah, there's one
Okay.
dead ahead right near that urchin.
Oh yeah,
Yeah.
on the right.
Yeah. That looks like it.
Oh,
yeah.
next
Up
to
there
the
right now.
urchin.
Center. Yeah.
I see him. You want me to zoom?
Yeah, let's get something.
You
Let's
can
see.
zoom.
Let's see if we can get this darker fish.
You're gonna get tugged.
All right.
Shape's right for McTophid.
Oh,
Yeah,
You might have to give it a minute or two,
that's okay. He's
or 30 seconds.
working with as much tether as he's got.
Yeah.
Yeah. Okay.
Hey, Chris. Awesome to know you've been watching the video every day. We saw a
That's it.
beautiful
Sorry.
hemicorallium last night at about 1,100 meters. Big, big gardens. And a bit of coral garden
Oh,
this
there he
morning, too,
Hold on.
just as we took over the watch, so about 2.5 hours ago.
He's really close. Oh, he's so close.
That shot's beyond my pay grade.
That's all right. No, hold on. Don't give up. I think he's...
Keep it going.
As long as he stays in front of us, which it looks like he's going to do.
Ah,
No, we're on
Nice
top
job,
of him.
though. Nice
We're on-
job.
Is that his brother?
That's his buddy, yeah. His brooder.
Well, okay then. All right.
All right, do you want to center up and then we'll summit
Yeah.
this cliff?
Okay. Bumping it.
Sure. I mean Jake, but
Yeah. Just letting him know it's about to shake.
Ooh, big
Oh.
fish.
Ruffy?
Ruffy.
That looks, yeah, interesting.
Cool
That
colors.
Looks larger than the ones
Yeah.
we've been seeing.
Look at that yellow coral,
What do
too.
you-
all right, we will need to start coming up.
All right, coming
Pretty
up.
Yeah.
sporty though.
Can
Yeah,
we
zoom on the
You
yellow.
zoom?
want this yellow?
Yeah.
Quick,
Yeah, real quick.
Real quick.
All right, that's all you get.
That's all you get.
So see on Argus there, we're 30 meters out from the cliff.
Yeah.
Yep.
Okay.
And because of the shadow behind it, you can see it's still vertical, even out of Argus's view. So that means we got to go up and find the top of it
Before you get drug over it.
before we get drug over it.
Yeah.
Legit.
So you might want to center yourself up too.
Yep. Okay.
Good call on the joy gain.
Yeah, Miles, can we start moving in steps so we have a little more control?
Yeah. It'll be slower.
Okay.
Cool rock texture here. You can really see the difference. If these were basalts, they would not have those pockets carved into them or sort of eroded away. Hello.
And we'll be okay. We got plenty of time. But if we didn't, you know how to kill your bias? The quick button to zero the bias?
I do not know that button.
It's right there.
Okay.
So you could undial it with the little dial as well, but if you need it in a hurry just to zero it out, which we don't need now, you can just hit that and you get an extra 30% or whatever up.
Yep.
This density of an enemy is really cool. You know, obviously a filter feeder sitting there with their stinging cells capturing things out of the marine snow and doing well in this area of lower oxygen. These rocks with their sort of pockmark are carbonate. They're part of the carbonate platform from up above formed. That Argus shot is just crazy. Look at it.
Really cool.
Amazing. So we are climbing up to the
Try
top
something.
of climbing up a mountain. Just think about it that way. The top of this mountain happens to stick out of the water just a little bit, and that's Jarvis Island. But this mountain is 5,000 meters tall. We've been climbing only
Is that
from
better?
about 1,800
Sure.
meters depth. But this is a massive mountain, and we're just seeing a little bit of it as we climb up here to the tip top. These carbonate boulders that have tumbled down off the side are formed when it was more shallow. So these are the ancient reef structures that you're seeing here.
This may actually be the base of the platform now that we're coming up.
What would that mean?
This is where the very bottom of where the coral started to form, the fringing reef.
Okay. Yeah, it doesn't look like they've broken in
No.
the same way.
It's a big sharp break in the topography too.
Those are like little crowns on top of these seamounts. Carbonate
Yeah.
crowns. Big fan over on the right. Starting to see them again.
The
We're doing steps now? Okay.
Trevor, can you explain what the challenge is when we're flying on something this vertical?
Sure, yeah. So the ship's moving along as we're going up slope. And if we come up to a vertical wall, the ship's still moving with the same horizontal speed, same horizontal velocity.
Mhm.
So we got to still make up that horizontal velocity while also going up a lot. So it's easy to move, 100 meters straight ahead. That's the end of your leash.
Yep. Put one in.
It's easy to move 100 meters straight ahead, but when you also have to go up 1,000 meters, for example, in that time versus going straight across, there's a lot more to it. So, yeah, it adds that extra little component.
I just got eye contact with Jake with the eyebrows like way up. Ah! That's a great explanation, Trevor. Thank you.
You're welcome.
That was a big cliff.
That was a big cliff.
Looks like that
Have you-
crevice might have been made by the last ROV pilot that didn't judge it right.
The previous intern.
They were just using their trenching lasers.
Yeah, speaking of which, there you go.
I love this question. In what way can viewers help support the work that we do? Let your friends and family know. Let teachers you know or students you know who might be interested in our work. Encourage them to tune in. Sign up for our newsletter. Follow us on Twitter at EV Nautilus, where you can get dive alerts. On Facebook, Nautilus Live. On Instagram, Nautilus Live.
Yep.
Just participate in exploration with us.
Yeah,
Yes. Go ahead, Rob.
Yeah, can we get over here and look at one of these coral over here to get a zoom on these?
Ship's still
Just to
moving along.
verify.
We'll have to stand by on that one for a sec, but
Okay, I
we'll
didn't
get
know-
there.
I don't know if we were just hanging here waiting.
You might have some leash.
We also have a donate button on the website if you're so inclined. Ocean Exploration
step.
Trust is a nonprofit organization. So a nonprofit organization owns and operates the ship and conducts all this exploration with a series of funders from grant-based and private donation funding. So if you are so inclined, check out that donate button or share it out.
Are those donations tax deductible?
Yes, they are. There's also the option to give monthly. So, you know, if you watch Nautilus Live as much as your favorite streaming service, consider us. You know, we're just so grateful and appreciative of the fact that millions of people around the world watch Nautilus Live each year, and that we get to share these stories with you. A big part of our work is not only to come and do this work, to understand that this urchin lives here and this coral lives there, and to help the science community know that, but to use the work that we do to inspire another generation of explorers and to help them understand all that's possible by, you know, by frankly doing your math homework, by considering a career in a STEM field. That you could come do work like this or whatever passions they are in your life that you want to follow. Cool Plagatharia.
How's the hand cramp?
It's good.
Nice.
Not bad. Too much adrenaline.
Bridge, can I have another step?
So Nautilus, viewers are confused whether Nautilus is stationary or not stationary. So we do not have the engines on. We're not actively driving around a whole bunch, but the ship is moving. Not stationary in the form of thinking about like an anchored ship just being in one spot. We are holding our position with a bow thruster and a jet pump, and then slowly marching along, sort of like walking dogs on a leash. If ROVs want to fly across a landscape or up a mountain in this case, the ship needs to move along with it. They only have so much leash in order to get out there. And Hercules is not ballasted or powered in a way to pull, you know, to really pull Argus a long distance. So this ship is taking steps. That's what you're hearing Miles communicate to the bridge, is what direction they'd like to move and how large of a step, you know. And then the ship uses the thrusters to push in that direction. So when we're tracking a line, we're moving continuously in one direction. When we stop and say we want to take steps, we hold position between them and then can make smaller, slower adjustments that are sometimes the more prudent thing to do when you're right up against a wall.
Oh, the steps are often a lot quicker than tracking a line. So it's a bit
Looks
of
like some
surge and stop and surge and
sea
stop.
stars, or are they slime stars there? Like sea stars. A number of them.
Oh, yeah. Some cool urchins, some coral rubble down there too. Looks like a dead one right in that hip of the valley, but big. Does Nautilus have an anchor? Yes, we do.
Oh, I hope so.
a little weird last night when sunset